In the 1970s, scientists achieved a biological milestone that contemporaries compared to the Apollo moon landing. They built the very first synthetic gene from scratch. To pull this off, a team of highly skilled chemists, led by Har Gobind Khurana, dedicated over 5 years of relentless work just to assemble a single gene. Every step happened at a physical lab bench. Scientists manually mixed liquid chemicals in glassware, coaxing individual pieces of genetic code to attach to one another, base by single base. While a spectacular proof of concept, this manual assembly line was far too slow to generate the volume of DNA required for actual medical treatments. Modern bioengineering relies on a continuous engine of discovery known as the design, build, test, and learn cycle. But in the late 20th century, this engine was choked at the build phase. Relying on manual chemistry meant producing raw materials took too long. Researchers could design brilliant theoretical therapies, but they couldn't physically iterate or experiment if they were stuck waiting years for their basic DNA building blocks to be manufactured. To keep the pipeline of medical discovery moving, the industry had to take the process out of human hands and move it into the realm of microscopic automation. The first major leap forward was solid-phase chemistry. Instead of letting DNA strands float freely in a liquid solution, scientists found a way to anchor them to a physical surface during synthesis, separating the growing strands from waste products. With the DNA locked in place, the biological industry was able to hijack manufacturing tools originally designed for computer chips. By using techniques like photolithography and programmable micromirrors, machines could effectively print DNA. This triggered a massive explosion in manufacturing volume. Where a team of chemists once spent years on a single handmade gene, a modern synthesizer can simultaneously print 10,000 distinct genes onto a single silicon chip. The financial cost of production collapsed alongside the timeline. Synthesizing a single nucleotide dropped from 15 cents down to a fraction of a penny. The printing constraints were gone, but gaining the sudden capacity to manufacture millions of therapeutic antibody variations instantly created a severe logistical crisis further down the line. Now that researchers could print synthetic building blocks by the millions, they faced the staggering physical problem of actually testing all of those experimental variations in the lab. Historically, testing relied on hybridoma technology, a method that required fusing immortal cells with mortal B cells from an immunized animal. This process was inextricably tied to the slow, natural pace of a living immune system. To match the speed of printed DNA, scientists developed a high-throughput screening tool called phage display. Researchers take the millions of newly printed DNA strands and insert them into bacteriophages, viruses that infect bacteria. This forces the virus to physically sprout the experimental antibody structure on its outer surface, allowing for rapid, large-scale screening against disease targets. This diagram illustrates the process of combining these printed segments. By combining printed DNA with viral screening, scientists completely bypassed the animal immune system. They could generate fully synthetic antibody libraries containing an astronomical number of possible therapeutic combinations. Today, biopharmaceutical companies operate at a scale that dwarfs those early discoveries. They maintain fully synthetic libraries packed with over 10 billion unique antibody clones. To handle this massive inventory, automated robotics take over. Liquid handling systems keep libraries in constant rotation, moving samples seamlessly through the testing pipeline. Yet even with high-speed robotics running around the clock, testing 10 billion liquid samples to find a single effective cure is an incredibly inefficient task. The physical testing phase has succeeded so thoroughly that it now produces far more data than human researchers can reasonably process. The primary bottleneck has shifted once again, moving directly into the final learn phase of the cycle. To clear this final hurdle, researchers are turning to deep learning and neural networks to make sense of these massive synthetic datasets. Instead of waiting months for physical lab results, scientists use artificial intelligence to analyze the library data and accurately predict how well a specific antibody will bind to a target, long before the physical screening even concludes. Trimming millions of physical tests down to a focused list of high-probability candidates directly accelerates the transition from theoretical sequences to viable physical treatments. Science has evolved from painstakingly assembling the code of life by hand to printing it on silicon. Now, by unleashing computational intelligence on that printed DNA, The next great therapeutic cure will be engineered and optimized entirely at the speed